Our report was based on the study of asymmetry and symmetry in brain waves from dolphin left and right brain hemispheres, some observations after anesthesia during quiescent hanging behavior and during visual obstruction. This study was done by Sam H. Ridgway from the Marine Mammal Program in San Diego, California. This study relates brainwaves of bottlenose dolphins to their sleeping behavior with the use of EEGs. Electroencephalograms are used to measure brainwaves and electrical activity of the brain. Studies about this sleeping behavior is very scarce and is poorly supported in previous studies. Learning more about cetacean brain activity during sleep can provide more comparative analysis between humans and marine mammals. One of the main reasons that humans study animal behavior is to learn more about ourselves and relate behavior to other animals. This study mentioned previous studies that hypothesized alternating sleep patterns in dolphins, but unfortunately neither were supported by EEG data. This study aims to support these previous hypotheses with statistical data. This study hypothesized that respiratory excursions in dolphins are incompatible with deep, slow-wave sleep of both hemispheres. Deep, slow-wave sleep refers to phase 3 sleep, which is also known as the deepest phase of non-rapid eye movement sleep. They also hypothesized that the left and right hemisphere of the brain would display different EEG waves. They want to provide more support for the other studies' findings that were mentioned in the research article. This desynchronization of the left and right brain waves should therefore indicate less activity in one hemisphere and more in the other during sleep to support the idea that dolphins sleep one hemisphere at a time. The purpose of this study is to learn more information about behavioral sleep patterns and brain waves in bottlenose dolphins during recovery from anesthesia, during sleep, and during arousal from sleep. Deep, slow brainwaves usually indicate dreaming during sleep for humans. Analyzing these types of brainwaves in dolphins can help make connections between the similarities and differences in cetacean sleeping and sleeping in humans. This study used electrodes to collect the dolphins' brainwaves. The electrodes used to collect the brainwaves required implantations because the skin, blubber, and muscle over the cranium were too thick. FM bands were recorded because they are associated with meditation and REM sleep. EEG waves were recorded and analyzed during recovery from anesthesia, during sleep, and during arousal from sleep. All data was recorded on a male dolphin that was placed in a small pool. The animal was only placed in a small pool for 7 hours at a time. This image indicates where the electrodes in the experiment were implanted on the dolphin.
EEG readings, represented in Figure 5, showed 5 to 13 Hz at 40 to 90 microvolts and 15 to 25 Hz at up to 30 microvolts. EEG readings also showed slow waves, about 2.5 to 6 Hz, with an amplitude of 240 microvolts while sleeping. Right hemisphere was reading as awake, while the right eye was open and the left eye was closed, while the left hemisphere read as asleep. The findings of this study show that there are marked differences between the waves in the left and right brain of the dolphin observed. The hypothesis was supported and results agreed with previous studies that created the basis for this particular study. Dolphins, even when awake, show lower frequencies of brain waves than laboratory studies done on humans and other animals. Slower brain waves with high frequencies, or amplitudes, usually means less activity. For example, when a person is sleeping, they show slower brain waves with higher amplitudes. This study covers 2 of Tinbergen's 4 questions. For causation, they asked what caused one hemisphere to quote-unquote sleep while the other was awake, and for function, they asked how does alternating hemispheres while sleeping protect against predators and things like that. For the bigger picture, this study adds to our general knowledge about cetaceans. It'll provide a better understanding of how to care for these animals when they're in human care at zoos and aquariums, and it could also provide useful knowledge for the treatment of human conditions as well.
Future research could include applying Tinbergen's other two questions, evolution and development. One suggestion could include the evolution behind sleeping behavior in dolphins by testing whether all dolphins or all cetaceans do this behavior and compare it to populations in the past. Another suggestion could include the development behind sleeping behavior by testing if this behavior is learned or instinctive. Do you think that this study provided enough accurate data about brain and sleeping behavior in cetaceans? Do you think there should be more data collected among different genders and different species of cetaceans in order to support these results? Do you think this type of experiment could be conducted in a more naturalistic setting? If so, how? What other studies could be conducted for future research?